Hi everyone. Yes, I really am Lindesay Scott-Hayward talking now. I'm from CREAM at the University of St Andrews, which is the Centre for Research into Ecological and Environmental Modelling. This is some work that we did for Marine Scotland, and the aim was they wanted some guidance on the statistical methods for looking at species distributions, in particular in areas of marine renewables. So the main aim of the project was to do a methods comparison so that we could come up with a particular method that was deemed to be the best one to use, broadly speaking, and then to develop an R package to make it available for the end user that might want to use it. If you want to know more about how we did the comparison and how we came up with the methods that we did, we're going to talk about that in the workshop tomorrow afternoon, the stats one. So we're looking at mapping spatial distribution from line transect or vantage point surveys, with our particular focus on spatially explicit impact effects. So this is looking at precisely where geographically we have changes in animal distribution, and if any of those changes can be attributed to devices, for instance. So previous assessment tended to measure differences in animal abundance prior to and following development, but this suffered from some disadvantages, such as attributing, it was difficult to attribute any potential change to a development as a causal agent, because all you've got is a difference in two numbers. You don't have any spatially explicit differences. Can fail to acknowledge other forces that might influence animal abundance and distribution, and is insensitive to more subtle changes in animal populations as well. So we present a new R package called MRSea, and I said it's developed specifically to tackle assessment of potential impacts of renewable developments, but could in fact be used for any kind of baseline analysis. And the functions we made up, we used to analyze segmented line transect data and or nearshore vantage point data. But as I say, they could actually be used in other scenarios as well. Oh, and see the CREAM website for details. That's weird. It's come up on a separate page. So what this was supposed to have on it was a nice little banner that says, we're giving a workshop on the 6th to the 8th of August this year on this package. So if anybody would like to learn more about it, then you can come and see. Oh, there it is. Look, fancy that. It's come up later. Oh well. So yes, we're giving a workshop. Come and find out how to use all these things. So the package itself, we thought that it would be useful to have example data sets. So you don't have to have your own data to work out how to use these methods. We have 6 data sets, 3 offshore, 3 nearshore, and 2 they have no impact effect. So we've basically got a before and an after scenario of no impact, decrease in general across the whole surface, and also redistribution of animals from one part of the survey area to another. The offshore data is in the form of line transect surveys and takes on the form, if you're looking for it in the package, it's dis.data. Either NO or DE or RE, depending on which scenario you want. And the vantage point is a grid of 41 repeatedly measured cells. Now, both of these are taken from they're simulated based on real data sets. So they're data sets that we've previously analyzed, and we've simulated off those to generate new data. And there's also a prediction grid for each data set, so you can make nice, pretty maps. So we have a variety of functions for fitting and diagnosing models, and most importantly, making inference based on those models. So I'm going to focus on the offshore survey data here with a redistribution of animals within a study region, just to show you some pictures of some of the things as we go along. It's basically a little show and tell, this talk. It's just to show you what's kind of there and available. We have observed counts with imperfect detection, and these were lifted from our simulated true surface. And in this case, we're using the dataset called dis.data.re within the package, if you're looking for it. So we have simulated densities of birds before and after impact. Some of you might recognize the region that we might have simulated this off. So we have an area here before. So before this is the star here represents our area of impact. So in this case, we're going to pretend maybe that it might be some wind turbines. And before, this is the distribution of animals across our surface. And afterwards, there's a slight decline around this area. And you can see that actually there's also a slight increase in this area to the far east. as well. So that's the true data that we've simulated off and given it some noise and made it over-dispersed and correlated and all that kind of stuff. So we use our distance sampling methods to adjust for imperfect detection. I'm not going to go into these at all because there's distance analysis workshops as well, and all of the distance functions we just lift out of the MRSea package in R, which is how you can do distance in R. We use something called create nhat to adjust our observed counts using the detection function that we've estimated based on the data. So once we've adjusted for detection, this is the kind of plot we might get. So the grey dots just show you all of the segments for our line transects. If it's grey still, then it's 0. If it's got a red blob, then it's got something in it. This is just a log of counts. So even just looking at the raw adjusted data, you can see that maybe there's less in this area and maybe there's more down here. But we need to do a little bit more than that rather than just eyeballing it. So we have over-dispersed counts. So we model them with a quasi-Poisson error structure. And we have a bunch of covariates: depth, season, impact. We have a spatial term. And we can also allow for a spatial impact interaction. And this is what allows for a redistribution in the surface. We use what's called a CRESS-GE framework to estimate the smooth terms in the model. And this allows for positive autocorrelation in our residuals. As you move along your transect, you have your data is correlated through time. So we need to allow for that. The CRESS part of this allows for our smooth terms, and we also have a spatially adaptive side to this as well, which allows a change in flexibility across our surface. So the functions, I won't go into detail because out of context they're not particularly helpful, but there's two functions for looking at how you fit your smooth terms to your covariates such as depth, so it's a 1D covariate. And you can either use this run south or 1D, which just looks at the best smoothness for each covariate, but doesn't say whether you actually want to keep it in the model or whether it should be a linear term. The other alternative, you can run it with removal, which cycles through each covariate and says, okay, well, here's my best smoothness, but how does that compare to whether it was actually linear, or maybe I don't want it at all? And so it'll spit you out something at the end, your model selection process, that says which covariates you've ended up with and how smooth they are as well. Then you get onto the two-dimensional side of things. So we have a smooth term of space, and we have another function called run south of 2D, which looks at how the smoothness of that particular spatial term. And we have a user guide. So on the Cream website and the software pages, all of this stuff sits. So there's a package available for Mac users, Windows users, there's a user guide, and then there's all of the guidance documents that went along with the contract that we did for Marine Scotland as well. The models that are returned are of class GLM, which means you can use all of the familiar things, if you're familiar with R, like summary, update, predict, fitted, residuals, etc. They're all available. The p-values, now you have to use a GE-based p-values because you need to account for the correlation in your data. So this is just an example of a table. Obviously, it would come out looking like R, not looking like nice PowerPoint, but this is the kind of table that you'll get out, and these are GE-based p-values, so you're accounting for your correlation in your data. Diagnostics, you want to know whether what you're fitting is actually a decent model. You've got things, partial plots, diagnostics such as plots of observed versus fitted. You can get marginal R-squared, concordance correlations, which are designed like an R-squared, but for GEs, because they actually constrain your results to be between 0 and 1. Fitted values versus Pearson's residuals, and you can assess these in all the same ways that you would assess models usually. You can also have runs profiles, which indicate how your correlation is through your data, and influence checks. So because we're doing GEs and we've got blocks of data, you can assess whether you've got any one of your blocks is really influential, either to your predictions or to your coefficients that you have in your model as well, and that's what these statistics do. You can make predictions using predict, because we've just got a GLM type model. And importantly, to get your inference, you bootstrap to get upper and lower percentile confidence intervals. And the key here is that you've got to combine your uncertainty from your detection process and the uncertainty from your the density model that you just made. So you have to get both sets of uncertainty involved in this. So there's a function called do.bootstrap.cross, and it calculates all of your new predictions, and it makes your bootstrap confidence intervals. So you can maybe do 500 bootstraps, you get a set of 500 bootstrap predictions out at the other end, and you can use those to calculate percentile intervals. Now, I won't show you the confidence intervals here, but the reason we created those is because we're interested in spatially explicit differences. Where on the predicted surface is there a significant change in animal numbers? And there's something called get differences, and that just takes all of those predictions that you made and looks at the before and after differences for all of your 500 sets, and establishes whether your interval contains 0, then there's likely no difference before and after. In this case, I'll just move on to the picture. So it's a little bit difficult to see, but the model knew nothing about where our site of impact was. Okay, that was just coming out from the data that we have. And you can see that here we have negative change. I don't know if you can see all the little dots, but those are the cells that were considered to be significantly negative. And over here we have significantly positive change. Okay, so if you're able to capture a redistribution within your study region, this is particularly useful, as you'll be able to find out where on your surface you have these spatially explicit differences. So just to conclude, this is an example of some of the functions available in the MRSea package and what you can do. A full list, obviously, is available in the reference manual. As I said, everything is on the website for CREAM. If you go to the software pages, you'll find the guidance documents and the package itself. We'd love to know if you have any problems, if anyone is using this, as we're always looking to improve it, and if you have any suggestions for what we can do to improve. We are running, we'll be talking about how we came about these methods tomorrow in the workshop in the afternoon. And also, just so there's a few references, but also just to remind you that we are running a workshop in August, and we'd really like it if you came. So that's all.
Thank you. Thank you, Lindsay. You could take one quick question if there is one.
It's probably too early for statistics.
I don't see one, so we'll move straight on. Thank you.
Okay. Oh, there's one more.
Sorry, Beth.
How would you use some of the data insights that are vantage point where you've got a lot of grids? What we discovered with the real data was huge amounts of zeros, 98% zeros. Does this deal with that kind of zero-related data?
So the question is about whether this will deal with very heavily zero-inflated data. So some of the zero inflation can be accounted for by covariates in the model. Other parts will be accounted for. This over-dispersed Poisson, the over-dispersion part allows you to deal with an overly high number of zeros in the data. There is always going to be some data which is so zero-inflated that you just don't have enough signal in the one. So the key is, if you've got a very high number of zeros, but you've also got some useful information in, say, where all your ones are, or all your counts are, if they're in one place, then that can tell you something useful. If it's just so dispersed across your surface, there may be cases where you can always, you won't have enough information in the data that you have. But we haven't found too many problems with being particularly zero-inflated in these cases.
Thank you, Lindsay.
Okay.
So,